watching The Daily Cell, and I'm your host, Chelsea Rodriguez, and this segment is After the Interview. Today we're going to be talking about the podcast Antimicrobial Eating Microorganisms and the Resistome with Quantum Dantas. If you don't really know what kind of podcast I'm talking about, I will leave the link to the podcast below in the description, or if you just want to look it up yourself, it's actually under the website American Society for Microbiology, and just click on Meet the Microbiologist and you can just type in the title of exactly what this video is. The interviewer of this podcast is Julie Wolf, and she's interviewing Quantum Dantas, and to get a kind of background of who he is, he's a professor at the Washington University School of Medicine teaching pathology and immunology. In the beginning, he just kind of explains how he uses tools from different synthetic biology techniques, and how his main thing is research on microbial communities, on how they live and develop. He's also very involved with human health and environment, and he talks a lot about how interested he is in microbes and how they live and how they work, and how he finds them so interesting and beautiful and just wants to understand how they develop and, just learn more from them. So his research is mainly about, he is searching for lignin-degrading soil microbes, and lignin is actually a major component of soil's organic matter. However, because it's such a complex structure, lignin degradation is actually a major occurrence. So in search of that, he actually discovers an antimicrobial compound while he's trying to do this research on finding lignin degradation. He finds that. Since the discovery, he's actually continued the study of resistome soils and the human clinical samples found, and try and find the similarities between the two. He actually finds it very important and useful. He talks about how very important and useful it is to actually collect background information from microbiome studies in order to form a hypothesis. You got to get more educated and do more research before you try and make a hypothesis or go more in depth into an experiment. Throughout his experiment, also, he has contribution from other scientists as part of his research, and he actually, what really stands out to him is the expression of a resistome. And a resistome is a collection of 4 type of antibiotic resistant genes and precursors of pathogenic and non-pathogenic bacteria. This expression was proposed by Gerard White, who is a biochemistry and biomed science professor, and he's a research chair in antibiotic biochemistry at McMaster University. Another scientist that proposes something to him and kind of helps him lead his way into this experiment is Julian Davies. He is a principal investigator in the UK, and he introduced the producer hypothesis. And he hypothesized that the antimicrobials were actually being produced from the same microorganisms, but they just needed to have some kind of protection against themselves. In this experiment, Quantum was expecting to find lignin-degrading soil microbes and use the antibiotic as a negative compound. But after collecting samples and observing them, he actually finds 3 in particular that contain life. Ooh, discovery. Where the soil microbes actually accomplished growth. So that was a major thing that he wasn't kind of expecting to find, which kind of led us into this another door. So now, from the beginning of the podcast, Quantum spoke about how he wanted to compare the similarities of the resistome between the soil and clinical samples, and he actually found that they are indeed very similar, but there's no he was unable to find like a guaranteed answer. There was nothing like bold, underlined print, like here it is, here's the answer. Sometimes, most of the time that's not the case for science. It's always something. There's got to be an explanation for everything. Quantum and the other scientists that were a part of his study actually continue on their research, focusing on the resistomes over time and how they form and transfer into the other microbial areas. Through Julia, towards the bottom of the podcast, if you scroll down a little bit, she kind of highlights the main things that kind of stood out to her, and the main question that Quantum and these other researchers have that they still don't have an answer to is that, do the genes move from the clinic to the environment or from the environment to the clinic? She also kind of concludes that the so-called context is the key to determining diseases, depending on how easily prone somebody is to a microbe and how it can affect one person and not the other. She uses the example of E. coli, kind of suggesting that the microbes in E. coli can cause UTIs in some part. An example of UTIs can cause that the E. coli can cause UTIs in some parts of the body, but can also have no effect in another part of the body, say if it was carried in the gut, as she uses for example. So pretty much what I got from that there was, if you have a disease such as E. coli or bacteria in one part of your body, you're gonna need antibiotics for something like that, rather than like say it was in your gut and we have these microorganisms that can kind of just fight it off on our own. So it just kind of depends on, that's what she's saying, the context. It kind of depends on the context of how prone you are as a person to different diseases or bacteria. Throughout this podcast, there was one thing that Quantum said that really stood out to me, and it also stood out to Julie because she quoted it towards the end of on the bottom of the podcast. And he said, "We think from a clinical perspective that antibiotics are these privileged compounds, but they're not really. They're organic compounds, and there are bugs out there that can detoxify these compounds and then eat them." So from that, I pretty much took that, antibiotics aren't always necessary. Of course we need them, but they aren't always necessary. It's just so cool and crazy to think about how our body is so complex and how, there's so many things that we can fight off diseases on our own, or even think about how, we can grow a human being and birth them out, and then here we are, like a whole person. It's just so crazy to think about. And this podcast, just after listening to this podcast, it actually really inspires me to look into more about what my body can do. And that's exactly what these scientists are doing. They're still, Quantum and the other guys that are helping him in his research, they're still looking into this and still looking into what else our bodies can do. And that's what makes microbiology just so interesting, not only in our body but in the world. Like how amazing and little tiny cells can affect your whole entire body, and how our own bodies can fight off these things on their own. So if you just want to look into more of this podcast, you can click on I'll leave the link below in the description. You can follow along or just look into that, but I'm definitely going to be looking into more about what's going on in my body, because that's pretty cool to learn. But this was the Daily Cell. I'm Chelsea Rodriguez, and thanks for watching. We'll see you next time. Bye.